And please feel free to ask at any time and anything. So again, that I really feel like, these days, when there is a will, there are many companies, and especially that, if you think with me right as of right now, that if you can think about like what are some of the questions biologically that we are currently feeling interested, and we felt like, actually that I don't know how many people are on Zoom. If you can unmute yourself and tell me, like, is there anybody who is in biology background? Is anybody in biology background? Or if you're not, perhaps you can say a no, so I kind of know that you are here.
I'm not in biology.
Not me.
I'm a physician scientist with a venture background, so love to hear more about what Marcus has to say. But yes, there's a science person here.
Great. And on site that Adam, he's in the biology field. So I just want to share, Michael. Oh, I see. It's how can I control that? Because this would be controlled by the touch screen. Sorry, we have a little bit of a technical difficulties here. So it is not on my own screen. No, it's just gonna keep on. Sorry. Through Zoom, yeah, that will be a better way to do it. So Michael, that here that it's this meeting is being recorded. It's I don't know how to delete it. Yeah, anyway, so we're going to continue here. So again, that if you're looking at here, that these are some of the questions that we are, as scientists, that we're continuingly to be interested. And I just want to give you some examples, like questions like, can we predict the next pandemic? Will there ever be a cure for the common cold? And again, that there are a lot of influenza vaccines are made companies here as well. And then can we design and manufacture medicines that is customized for individual people? And this is one of the concepts that is getting ever increasingly popular as we call it precision medicine, personalized medicine. And of course that we have this, are you finding this interesting, that can a human tissue or organ being fully regenerated? And basically that how is homeostasis? Sorry, I'm hearing my feedback. If you can unmute. And how is the immune homeostasis is being maintained and regulated? And again, that there are questions on, is there scientific basis on the Chinese medicine? And again, that back then, if you remember, the Chinese for the a lot of the Chinese traditional medicine extract, it seems to be very potent on specific disease. Can we find a scientific evidence on these? And then how can the next generation of vaccine be made? So again, that for the longest time that it has been this empty capsids of the virus, and then, or we call it dead virus to make the vaccine. But now it's the messenger RNA vaccines, but can we engineer the next generation, right? And also, what is the etiology of autism? Because now that we have ever increasing of understanding of autism, but again, that how can we better characterize it? How can we better treat it? And eventually, how can we kind of eliminate or, all these questions? And can we overcome the antibiotic resistance? Because again, that there's this ever increasing opinion on if we just keep taking antibodies, that it might be one day we will develop full antibody resistance. No antibody will work on us anymore. And the list is just going on and on. And again, I just want you to know that all these questions, that there are companies and there are multiple companies. And today that I just want to share a little bit of each, a little bit of a company, what are they working on and potential targets. And then hopefully I can give you guys some ideas on, again, like what will be the next trend. So the Bay Area, basically that it includes San Francisco, East Bay, and of course the South Bay. This is literally 1/100th of the company listed. So again, that all these companies that they are from small, and there are even stealth mode of these biotech startup companies. So everyone that it's working toward all these scientific questions, everyone is interested. And again, that if you're looking at here, that specifically, if you're looking at, so the Bay Area as being the top of the total life science employment. And I want you to focus on the California life science, not the wages, but the sectors. So as you can see that there are academic research. So these academic research are the driving force for the vast majority of the startups. So most often it's these really big labs that they would have grad students, postdocs, or specialists. They would discover a potential target or an inhibitor or a specific technology, and then they will funnel into biotech. And of course, that there are matured biopharmaceuticals, and then also biorenewable companies. And then there are these software and hardware for medical device, instrument, or diagnostic companies. I will give a couple of examples later. And then there are these pure R&D testing labs. So again, COVID testing are one of the best examples back then. With the pandemic, it really made a lot of these testing labs. And of course, that there are also wholesale trade, these type of companies that I'm not very familiar with, but of course, it also exists. So overall, that again, Bay Area being in California and really has a lot of opportunities in terms of talents, including UC Berkeley, UCSF here, as well as Stanford. So it really provides again, the talents and the workforce, and of course the ideas to advance the next generation of health and of course the medicine development. What I want to do is I want to give a little bit of introduction about myself based on my professional training, because in that case, I think it will be a better way of sharing with people what do I know, because again, biotech is a big field I want to share what my training is, and perhaps that if you find that some of the directions that interest you, I can definitely elaborate more. And then if it's not necessarily my training background, unless that company is really famous, like Google, that I may have a lot to say. Other than that, I might have limited information. So I am from. I'm from China, and here is the province that I'm from, Liaoning province. And again, I'm from northeastern part of China, and a small town of 4 million people. And I came to the US. I had my undergrad in Shenyang Agricultural University, and back then that my major was biotechnology. So I know a lot about a little bit of everything, microbiology, plant physiology, soil science, and then light microscopy a little bit, and then developmental biology, zoology, botany, all these, only just a little bit of everything. And it was really that I want to start having a focus, and that's when I came to the US. And then back then that my advisor, UJ McMahon with Mark Harlow, they were moving from Stanford to Texas A&M University, and that's when I followed them. And my major back then was neurobiology. And I was focusing very specifically on a site. So this site is called a neuromuscular junction. And then this site that is important in terms of disease is a myasthenia gravis or spinal muscular atrophy. So these are the parts that it's really important. But instead of looking at the disease mechanism, I was just looking at what is going on, what are the normal physiology at that site, which is called neuromuscular junction. So here that if you're looking at the orange dot, so those are called the synaptic vesicles. And apparently that with the advancement of light microscopy, this is almost a decade ago, is when super resolution microscopy really started to take off. And then a lot of light microscopy compared with transmission like electron microscopy really allow us to look at, this is a very small structure. And we're talking about nanometers or as big as microns. So I did a lot of protein chemistry. So I know a lot of structural biology as well. That was also the time that potassium channel crystallography, crystal structure was discovered. And again, that was discovered by Brian Kobilka at Stanford University. So again, like that was the time, and RNA-seq just became a technology, a fancy technology, whereas right now that every lab basically can do an RNA-seq. With the basic knowledge, that's when I started to want to focus more on a little bit more related to human, because back then that the neuromuscular junction is actually conserved in a lot of vertebrates. So we're using mouse and we were using frogs neuromuscular junction. So I want to get a little bit closer to human biology and physiology. Some of the major discovery within my lab, and my advisor was the department chair of neurobiology at Stanford back then. So just by using transmission electron microscopy to look at synaptic vesicle structure, we called it ultrastructure because it's a higher resolution, and also to discover what's inside of the synaptic vesicle. So these are the major discovery. So I wouldn't go too much into details, but it was my 6.5 years of work. I can talk for days if you are interested. So with that I came back to Stanford, and this was, I was joined, supervised by Lu Chen and her husband, Tom Sudoff, who back then just got Nobel Prize in 2013 for discovering basically the synaptic vesicle recycling cycle. So that's why that I know. And Lu Chen's study back then is called the synaptic homeostatic plasticity. So this phenomenon that it's important for pediatric epilepsy or autism as well. So I was interested in disease, but I'm interested in a little bit more of a disease mechanism. So I joined the lab, and then that was when I get into, so that was back then, was 2015-16. That's when I got into the CRISPR because it was discovered in UC Berkeley, well, more of utilized at UC Berkeley by Jennifer Doudna, and then also University of Umia with Emmanuel Charpentier. And of course, that meanwhile, that at Feng Zhang at the Broad Institute at Harvard was parallelly working on similar. And that was also when I started working on induced pluripotent stem cells. And then specifically, I was using induced pluripotent stem cell, inducing it into human neurons. But back then, that all the other cell types that I was getting from either rats or mouse. And then I learned a lot about molecular cloning. So it is a process to get specific sequence out of an organism. And then also that a lot of the cell line engineer, can I have a protein to express that it's exogenous in the cell line that I'm interested in order to study the protein, or maybe in order to visualize the cells. So that was, I did a lot of those works. And of course that I did a little bit of neurodevelopmental disease modeling, which is autism disease modeling. And again, that's, again, as you can see that I'm walking away from the basic science and getting closer and closer to medicine. And that's when I really look at UCSF, that they have way more people who are focusing on clinical work, and specifically for Alzheimer's disease and frontal temporal lobar dementia, because UCSF has a really big Kavli Institute for Fundamental Neuroscience, as well as memory and aging. And UCSF has one of the largest cohort in terms of longitudinal patient tracking, and for Alzheimer's and frontal temporal lobar dementia. And I decided to join at UCSF here. And I was supervised by Eric Yulian and Eric Huang. That's when I really started utilizing CRISPR. When I was at Stanford, it was more of just looking at one gene. But at UCSF here, that we're looking at hundreds, if it's not thousands of genes. And then from focusing just to one cell, which is neuron, which is the cells in the central nervous system, if you know that if there are billions of neurons in the brain, and there are trillions of astrocytes and microglia, glial cells. So I started to learn how to induce iPSCs into astrocytes and microglia, and then really focusing on frontal temporal lobar degeneration and Alzheimer's. And back then at Stanford, it was a while ago, that there was a scientist called Ricardo Domecq. And before at Stanford, he went to, after Stanford, he went to Novartis, became the VP of R&D, and now he's working on something else. But it was really him, and later on was Sergio Paschka, and with Anka Paschka currently are still at Stanford. They started this, they call them mini brain, which is basically using iPSCs to induce into a small tissue that they behave just like a brain. So this is a great system because that it recapitulates a lot of things that human brain have. For example, the cell complexity and the activities, a lot of electrical activities going on. So again, this is a really good system, and cerebral organoid culture becomes one of the some of the better systems to model human disease, because before this, a lot of people are using mouse brain, rat's brain, and monkey brain, but no matter how close they are, they're not human brain. A lot of cell types are different, and a lot of the cell types that rodent or even other type of mammalians, they don't even have a specific cell type. So cerebral organoids is really a good advancement in terms of disease modeling. And again, that's looping back, that's when Jennifer Doudna finally got recognized by her kind of a discovery of CRISPR. And then it was the 2020 that she got Nobel Prize and shared with Emmanuel Charpentier. And again, that I joined this organization, I will talk a little bit more, that it was really spearheaded with the GSK and UCSF and UC Berkeley. And if you know, there is another person involved called Jonathan Weissman. He's the person discovered CRISPR genome-wide screen. So again, CRISPR is a technology, but to apply it to the next level, it's called a genome screen. And then now that I expand my repertoire, that I started focusing more on immune-related disease, including lupus, because that a lot of lysosome in the cell seems to not being functional well in specific cell type like macrophage or microglia. And that's why that part of my disease focus go into lupus or lysosomal storage disorder, also Niemann-Pick disease, as well as Hunter disease. So they all seem to share lysosomal storage disorder phenotype. And also that recently, for the last 2 years, that again, I recognize that UCSF also have a big consortium of aging tumor as well as a pediatric brain tumor. And I was basically inspired by Michelle Mungy when she was at Stanford, when I was at Stanford, that back then that midline glioma was just came to kind of a prognosis and diagnose, and it's a devastating pediatric brain tumor. So I was very captivated by this, and now I'm trying to use iPS-induced cerebral organoids to model the brain tumor interaction with the brain tissue. And then now that a lot of technology needs to be employed to study these highly complex disease, and as well as within GSK that there are a lot of resources provided. So I can do things that it's like a spatial transcriptomics, which is a highly technical and it's a time-consuming of a preparation, as well as not just looking at RNA-seq of, we called it bulk-seq before. So it's you pull all the cells together. You put, this is what Aviv Regev, who was the discovery for the single-cell RNA-seq, she was at the Broad, and now she's at Genentech as the VP, that she's using smoothie as the analogy. Before that you would just have all kinds of fruits, you blend it as a smoothie, and then you try to tease out, is there strawberries or bananas inside? But now what we can do is we would just look at this as a Lego parts, that every part that you can tease out and to identify what exactly cell it is. And then also that with small labs that you're not really able to automate things. Again, a lot of these genome-wide library that we're talking about plates, we're talking about hundreds, if it's not thousands of plates. And again, for scientists, I can speak for myself that the way how I work at 11 are different compared with 7 p.m. So again, but automation can work unexhaustively, right? So here that these are the technologies that I'm familiar with. And with all these things that I kind of learned and worked on, that I want to share a couple of examples of the companies that they are doing the things that I'm working with directly or indirectly, or on the disease that indirectly or directly that they're focusing. So the first one that I want to point out that, Genentech, this is such a unique company. Again, I'm going to go from larger size to smaller size. Genentech originally started by several people at UCSF, and then they allegedly bring the formula and started a company, and there were lawsuits followed up, but basically they are the largest manufacturer producing human synthesized insulin. And then there was this dwarfism, so it's lower, shorter than the average of a human growth in height, that protropin is their other kind of a star drugs. And on 2017 that they started this drug that it came to market, that this is for treating MS, multiple sclerosis. So what I want to say is that they spend an increasingly and just so much more effort and capitals on neurodegenerative disease. As much as currently there are not a lot of drugs on the market by Genentech, we're hoping probably that in the next decade that there will be new drugs, hopefully discovered by Genentech, can come to market. So Genentech is definitely, again, for neurological disorder, they are definitely really one of the biggest effort in the Bay Area or in the world. And of course, that recently acquired by Roche. If you know Denali, Denali originally, if you remember, when we think about Regeneron, we think about Mark Tessier-Levine, which is our president at Stanford University, and he was actually one of the co-founders for Denali. And actually a lot of the people at Genentech, because if you know Mark Tessier-Levine, that he was at Genentech before, so several people from Genentech, including Ryan Watts or Joe Lukoc, and they all left Genentech and started this Denali, which is very much neurological disorder focused. And I've been to a lot of conferences and hear them talking about their drugs, or they also, yeah, by the way, so Genentech is different, and they're definitely leading the trend in the sense of even being a biopharma company, they very much focusing on peer-reviewed paper publication. So which really give them a lot of academic credibility. And in that sense that they attract a lot of scientists from UCSF, UC Berkeley, and Stanford. And Denali, following what Genentech is doing, only that they don't do as much as what Genentech doing in the cancer field. They're only focusing on neurological disorder, and they're also focusing on publishing as well. And some of their targets are, again, these are gene names, LRRK2 or TREM2. So those are the gene's name. It seems to be a very ever increasing evidence that it's pointing to this might be the target to cure neurological disorder, including Alzheimer's disease or frontotemporal lobar dementia. And what Denali is doing also is they have their in-house discoveries for basic research, but they also collaborate with different labs across the world. So again, one of the collaborations are with WashU St. Louis and also grew from Germany. So one of the faculty that I know, his name is Christian Haas, is specifically working with Denali on the TREM2, this target. And then TREM2, I guess, also collaborates with WashU St. Louis. By the way, that WashU St. Louis has really good neurological disorder kind of a field or a neuroimmunology field as well. And then again, that they have, again, they seems to have compound lined up for Hunter disease, which is again, lysosomal storage disorder. And then they develop new technologies like how to get particles, like small particles, and go across blood-brain barrier. Blood-brain barrier, if you know that it's the kind of, we call human brain our immune privileged, compartmentalized of a system. So again, a lot of drugs that you can take whatever you want, but it's just not going to go through blood-brain barrier. So that means it's going to go to waste. So again, that a lot of the technologies are actually going through how can I get my drug of interest across blood-brain barrier? So this is one of the efforts that Denali is working on as well. And then there is another company called Elector. Elector, this CEO was really for back then at Stanford. There is a faculty called Ben Barres, the late Ben Barres. So he or she was the really the driving force for people to recognize glial cells in the central nervous system. And therefore that, in the central nervous system, in addition to neuron, there are glial cells. There are majorly three types of glial cells. Astrocytes, that it's important for interacting with neuron, helping with neuronal development. They can also help with neurotransmitter recycling. Oligodendrocytes. Oligodendrocytes are important for myelinating the neurons. So a lot of MS or fibromyalgia, all these are due to oligodendrocytes are not myelinating anymore. It's like your wire, your electrical wire, those insulating of these rubber. So that's what oligodendrocytes is doing. And then you have microglia. And microglia seems to be important for neuroinflammation. So from the very for the last decade, that what the effort was basically showing amyloid beta and tau protein tangle, that it's the hallmark of Alzheimer's. But now that the idea is completely shift into, it's because of the glial cells seems to become pro-inflammatory. And that means that when they go crazy, they just keep killing all the cells in the brain. So again, Elector is one of the first company to just really focusing on glial cells. And the founder, Anand Rothenthal, that he really, goes to a lot of embarrasses, lab members or symposium, like everywhere that you will see him, that he's the founder of Elector. So again, they're focusing on TREM2, they're focusing on FTD, frontotemporal lobar dementia, and Alzheimer's as well. And that the company gets smaller and smaller and smaller. Elitarian, this is one of the most recent one I came across that because I talked with their scientific director, Tracy Yuan. She was trained at UCSF here with David Rowich. again, studying MS. And again, as you can imagine, 2017, this MS drugs, she was at Genentech as well. So I would assume that she knows a lot of these program or project related to MS discovery. And she kind of has her own ideas, and she was talking with a lot of VC firms, and hence this Litarian, this company. And this company is specifically focusing on MS-related drugs, and also the maybe glial cells kind of focus. And they did pull a lot of people from Genentech, Denali, Alector as well. So, and meanwhile, that in order to study neurological disorder, there are a lot of artificial intelligence and machine learning companies are coming up, because that the way how we study science, on one hand, these old-timey Genentech, Denali, Alector, Litarian, that they may or may not have their individual target, but a lot of companies wants to ask, can I get 5, tens, or hundreds of targets that I can go at the same time? And that's what AI ML comes in. So in vitro, I think she is a Stanford professor, Daphne Kohler. She was also the co-founder of Coursera, if you know. So she co-founded the company called In Vitro, and they are focusing on, among some of the directions, they're also focusing on neurological disorder. Herophilus is another one that they are, their cell is really about cerebral organoids modeling, high content imaging. So if usually I can only look at one organoid or half of an organoid, but again, Herophilus is developing, can I look at thousands of organoids at a time? So that's what they are doing. So a lot of these neurological disorder companies are kind of, I wouldn't say old-fashioned, but they're very focused. They focus on a disease. If they have a gene of their interest that they go after, and then their strategy is more of like, I want to collaborate with the best in the academic lab. So these are a lot of these, they do their own R&D, but they also collaborate academically on specific things. So this is kind of their way of discovering and how they organize. And then we have the induced pluripotent stem cell companies in the Bay Area. BioMarin, the company was never in my attention at all, that they recruited a couple of years ago, Kevin Egan. Kevin Egan is really famous. He was a Harvard fellow at Harvard, and then he became like a very fast, like usually it takes decades promoted from assistant professor to PhD. I think he got promoted like within 5 years, and then associate to. So his research is again on ALS as well, amyloid lateral sclerosis, ALS, and then also a lot of lysosomal related disorders from his lab. So I follow his lab, and then one of those days I know Kevin Egan quit Harvard and joined BioMarin, and that's when I really start taking BioMarin more seriously. And then now that some of his trainees, former trainees, including Justin Cheetah, he was at USC as a faculty, he joined BioMarin as well. And then some people I know from Denali also recently joined BioMarin. So I'm paying ever increasing attention to BioMarin, and I encourage you to pay attention. And again, one of the things that they talk about publicly is, again, to treat dwarfism. And they recruited a lot of people to study genetics. And again, at Harvard, there are a lot of labs trying to study bone development in order to cure dwarfism. And again, as I mentioned, that Genentech is also having a drug, protropin, that it's related to dwarfism. So again, can they treat this? So this is the disorder that BioMarin is really talking about at conferences. New site, this is Stanford faculty Marius Vernig with Tom Sudoff, which was my previous advisor. New site is specifically because, again, that it's really hard to generate IPS-induced neurons. So somebody needs to make them for the researchers. So this company is mainly to do, if you have induced pluripotent stem cells, I can make it into neuron. I can sell it to you. You can do your experiments. So this is kind of like a service providing of a company. By the way, that I don't know if you guys are familiar that UCSF or even Gladstone, Shinya Yamanaka, which won the Nobel Prize for discovering how to change somatic cells with transcription factor, turn them back into induced pluripotent stem cells, because otherwise that we can't study it unless we're using embryonic stem cells, which are way less accessible. But now that with these transcription factors, that we're able to turn any somatic cells from any patients into the induced pluripotent stem cells. And then I can induce it into astrocytes, microglia, any cell type of interest. So NewSite is the company that they're making the cells for the researcher. What do you need? Oh, you need this patient background, you need neurons. How many do you need? Oh, I need 1 million. There it is. And it's like 50,000. So that's kind of what it is. And again, Marius Wernig and Tom Sudoff, they were the first one discovered that NGN2 is called neurogenin2. Actually, one works well as well, can turn iPSCs into neurons. So again, they have the major discovery, and then they can make it reliably, and then they can have a company. That's kind of what they did. Fujifilm. So Fujifilm started dealing with iPSCs as well. So Fujifilm, so his biggest cell is iPS-induced microglia. So I learned iPSC-induced microglia with the original author that I went to UC Irvine to learn that the author name is Matthew Blurton Jones at UC Irvine. So I went there to learn how to induce it. And again, this is a really difficult cell type to induce. But again, a lot of needs. People want to know how to do it. So there comes Fujifilm. If you don't know how to make it, I make it and I sell it to you. So again, this is a little more of the service-based. And also, if you have a gene of interest, I like to have a GNA knockout of a microglia. Okay, I can make it. So that's kind of, again, it's a service-based of a company. And then there's another company called Brain X Cells. So this company is a collective of all kinds of cell types that they can induce. May or may not be the best way of inducing it, may or may not be the best quality, but they can induce it. At least that you have something to work with. It's better than nothing to work with. This company is not in the Bay Area, but I want to point it out. And then they can induce different cell types for you and sell it to you. Neurona, this is founded by UCSF faculty Arnold Krickstein. And this is, they say that they want to study neurological disorders. And I have yet to see the product. But again, they are attracting a lot of attention. And they're not releasing any drug targets or products, but Arnold Krickstein, this professor at UCSF, it's very well known. He was former chair of Eli and Edith Broad Institute for Stem Cell and Regeneration Medicine. So with his last 10 years of being a director, that the UFO building, which is just behind me, that it was established, and then the large endorsement of the Wiles company, the Wiles family, as well as the Broad family. East Coast is the Harvard Broad, and here we have this, and as well as the UCLA, the Broad Institute. So again, that a very successful faculty study brain development. And again, his company is perhaps it's iPSC related to treat disease. But again, I just know they use iPSC, exactly what they do. They're not publicizing it yet. This is a New York company. I really want to point it out because that if you think, when is the stem cell transplant can happen, I can tell you that in peripheral to treat leukemia, beta thalassemia, those are actually going. It is. It was started with This was started with the IGI, Innovative Genomic Institute at UC Berkeley, and then the first ever transplanted patient, her name is Victoria Gray, and she was being transplanted. I think she has terminal leukemia. and then she got CRISPR edited HPC, which is iPSC turned into HPC, so her hematopoietic stem cells to generate red blood cells, and then xenotransplant into herself back again. After a year, she's still alive and kicking, and then you might think that, oh, it'd be really impossible, or when would it be the time that we can do xenotransplant into human brain? Actually, that it happened already. So this is why I bring Blue Rock in here. So there's a faculty at Memorial Sloan Kettering in New York. His name is Lawrence Studer. So he studies, again, iPSC cell fate and also a lot of disease. And his lab is, again, amazing discoveries almost every year. And one of the things that he's working with neurosurgeon to treat the late onset of Parkinsonians, so Parkinson disease. Because again, if you remember that Parkinson's has these tremor, and again, it's mostly due to the substantia nigra dopaminergic neuron massive cell death. And again, these type of neurons are really hard to regenerate, and then let alone to xenotransplanted. But again, that they've been recruiting volunteers, and then they've induced the dopaminergic neuron, and they inject into the brain of the patients, and they actually see beautiful integration of their cells with the brain, within the brain, and it seems to elevate some of the symptoms of the tremor. So again, like this is like a big major advancement. And again, that they are going on phase 3 right now. So phase 1 and 2 are amazing. And again, that as far as I know, only this one company that it's doing the central nervous system xenotransplant for iPS-induced cell types. So that's why I want to bring this to everyone's attention. And of course, that is the genome editing, the technology. With 2020, there, I don't know if you guys familiar, that the last decade or ever, scientifically, the biggest lawsuit, it is the CRISPR lawsuit. So there is a book I encourage, if you are interested, you should read it. It's by Walter Isaacson, that he wrote a biography for Steve Jobs as well. Walter Isaacson wrote a biography for Jennifer Doudna after she got the Nobel Prize. It's called The Code Breaker. And basically, they were talking about the history of CRISPR technology. The CRISPR technology, and again, that it is a real-time war in between the Broad Institute of Feng Zhang and as well as Jennifer Doudna with Emmanuel Charpentier. The biggest controversy is who discovered it first and who was the first one putting into eukaryotes. So CRISPR, this kind of a system, it was discovered in prokaryotes. So it's in microorganisms, and people look at this as one of the microorganisms' immune defense. The system is that they would survey the actual foreign DNA insertion, and then the bacteria themselves can cut the sequence, and then prevent the sequence to be integrated. And this was the discovery, and that's what Jennifer Doudna and Emmanuel Charpentier discovered and applied. But what really is that to test, just because it's working in prokaryotes, doesn't mean it's going to work in the eukaryotes. And that's when Feng Zhang came in. Feng Zhang firstly demonstrated that actually it's working in eukaryotes. And that's when the technology really exploded, because if we can use that in that way, it is rewriting our own genome. We can play God ourselves. We can just go in ourselves and cut and paste all of our genes. And that's when both the Broad and the UC systems see the profits. So the whole war that it has been, it's a patent war. Patent war lasts for a very long time. But nevertheless, that the scientists, they are advancing the science and discover a new genome editing technology. Before that, we called it RNA interference. It was a lot of off-target effects, not very efficient, a lot of cytotoxicity. But with CRISPR technology, that it was higher precision, but still not 100%. So it's a higher precision. And then if you have a guide sequence, you can pretty much modify any genes you want. So again, that this is really expanding. And of course, all these VCs see the potential of this. And that's why, and the lawyer and all geared up. And this, and what I want to say is now that if you can see there's a West Coast and East Coast, if there is a West Coast company, there is an East Coast company because it's on CRISPR and it's on almost a similar technology. So here, the first one, graphite, this was founded by one of the Stanford faculty, Matthew Porteous. He specifically studied hematopoietic stem cells, again, beta thalassemia, leukemia, and pediatric leukemia. So that's what Matthew Porteous is doing. And again, his mainly focus is on how safe it is to use on human. So again, just because you think it's okay, we can't use it on human, but how safe, and therefore this graphite company. And then for the mammoth and caribou, so both of these two are Jennifer Doudna's company by her former grad students or postdocs, and they are both leveraging the CRISPR technology to treat all kinds of disorders. And now that Jennifer Doudna publicly talking about that she's interested in the next generation of, I don't know if everyone's familiar with the idea of a microbiota or our microbiome, that it's in the gut, that it's in our system, especially the digestive tract, and can they be the remedy for a lot of things? Because recently that, we know a lot of these bacteria, all kinds of bacteria, they secrete whatever it is, it seems to be helping human. And one of the things I find it quite interesting is Parkinson's disease patients who usually have constipation. And we don't know if it's because of again, some of the microvilli, those cells that some people, they claim, some labs, they claim it was because of their calcium channel, potassium channel, that it's malfunction, or it was because of the microbiota in the Parkinsonians' gut, that it might secrete some kind of a toxin that it's exacerbating either Parkinson's symptom or the constipation. But again, I'm really excited to see that there are more microbiota companies and to think in the way, not just probiotics, but more of treating some other disease as well. Sangamo is this company that, again, they started with the, again, before CRISPR, it was RNAi. There's another thing called zinc finger nuclease. It's like before scissors and there are knives, and there are also some stones that can cut. You just want to cut the DNA, so they're less and less efficient. So Sangamo was one of the companies that they discover some sharper stones. But again, you have to sharpen the stone first, whereas that a scissor company can immediately just, again, you just go in and cut. But Sangamo, again, that they're still focusing on genome editing, and they know very well with the CRISPR, and this is the company as well. Synthago. So again, with the CRISPR technology, again, it cannot be just CRISPR to patients. So what they are doing is that they would go CRISPR, someone else who studies CRISPR. But again, who can help them? So Synthago is a kind of a researcher facing of a company. Where do you want to cut? I'll go and cut it for you so you can study what it is. So that's kind of what they're doing. So they help you to they help us at least, personally speaking, that we have a lot of things from Synthago. So they will synthesize a nucleotide or sequences, and they will help us generating knockout. So again, this if I use CRISPR to modify the gene, it's easy said than done. So it's a very time consuming. So it will take a lot of time, and that's why we need someone else to do the dirty work so I can study it immediately. So Synthago is doing the dirty work for researchers. And of course, they charge a lot of money. Tune, again, this is founded by Fyodor Ornov. So he is a professor at UC Berkeley. And again, that he's studying RNA modification, DNA modification for decades. So again, that he being the co-founder, that it's really, again, thinking about CRISPR and thinking about everything. So this is, again, he is one of the co-founder for Tune, again, genome editing. And Scribe, this is also one of Jennifer Doudna's company. So again, a lot of the CRISPR, so you can see that there are way more examples and way more new startups with a new technology. So again, if you want to invest for the next thing, you just stay and put. If there's a new technology, that's when you invest. So that's kind of this how it works. And again, for East Coast, that there are all these companies that it's parallelly. Again, that there are all kinds of taking advantage of CRISPR and they added it. So a lot of these. And the co-founder, in addition to Jennifer Doudna, Feng Zhang, or even Emmanuel Charpentier, so there is another person involved. His name is David Liu. David Liu was recently elected as a member of the Academy of Science, and he's at the Broad, and he discovered that instead of you can do knockout CRISPR activation inhibition, you can also use base editing or prime editing to even more precisely edit it. So of course, that's the calling for a new company. So that's what it is. So again, that this is CRISPR technology really started so many companies in both West Coast and East Coast. Toward the end that I have just 2 or 3 more of a quick kind of I'm not going to go too much into details, but now that there are also, you know, all these trillionaires that everybody wants to live forever. So you know, again, that for the next 100 years, we will be still interested in why do we age? Or in other words, is can we reverse or stop aging? So there are a lot of age-related companies. So one of them being, so this was a great story. So back then that Hall Barron was the chief scientific officer at GSK. He was approached by Jeff Bezos first, and then among other investors that who wants to live forever. And then they want to start a company of studying human aging. And then immediately they pulled several faculties specifically from UCSF. Peter Walter is one of the faculties, Susanna Rossi, Hannah Al-Samad. So all these faculties were pulled, and now that they joined the Altos. So this company is called Altos. Currently, they're very well funded, and it's projected to have 4 centers. The first center is being at the Bay Area, and specifically with Peter Walter, Susanna Rossi, and Hannah Al-Samad. And basically, they want to leveraging AI/ML and, of course, larger drug screening or functional genomics to study what are the contributors or genes or cytokines that is important for aging. And then that in Kyoto, which is led by Shinya Yamanaka, and again, to study how can they do it from the perspective of cellular level. And again, that they have a site in Tokyo or generally in Japan. And then there's a site in San Diego, which are exactly, I don't know what are they doing, but it might be small molecule or looking at specific structural protein and a lot of computational. So a lot of people, and that part was led by Juan Carlos Isbia Belmonte, and he was at the Salk Institute, and then collaborating with other faculties, because again, UCSD, Salk, Stanford Pribby, this is another hub, or even the Scripps. So it's another hub. So again, they are hoping to address. So Altos. And then Google has 2 spin-out for aging-related companies, and they're both pretty big. The first one is called Calico. Calico was one of their scientific officer, or their chief scientific officer. Her name is Cynthia Kenyon. Cynthia Kenyon was at UCSF. She was a faculty. She discovered several genes in nematodes, or basically worms, that there are some genes that is related to aging. And she was recruited to Calico to lead a research program is, can we do screens? And currently a lot of the screens that they're published, it's on yeast. So it's East Sacramento. So it's a little bit primitive, but we haven't seen anything that it's done on human or a little bit higher of an organism. So that's what Calico. And recently they recruited Oliver Hahn from Tony Weiss-Corey's lab. Tony Weiss-Corey is at Stanford. He specifically studies aging, and he was trained with Thomas Randall. And so again, like they want to study human aging from different perspectives. And Verily is a strictly confidential of a company study aging. I have no idea what they're doing, but I know there's a company. They make you sign a contract. Was it 50 years? You can't talk about what you do. So this is but I want to bring this to everyone's attention. Gordian Bioscience. At the Bay Area, we have UCSF, UC Berkeley, and Stanford, but there's another place called the Buck Institute. Buck Institute is at Novato. So Eric Verdin was a faculty at UCSF, and then he was recruited to be the chair or the president of the Buck Institute. Buck Institute is specifically study aging. And then there was a postdoc, his name is Martin Jensen, I think his name is Martin Jensen. Don't quote me on this guy. But again, he co-founded the Gordian Bioscience, and it is an aging company, and they're being very active on scientific communications, and they want to publish, they want to work with other labs. And recently that I see that they're trying to organize happy hours and get UCSF postdoc, perhaps for the recruiting purpose, or maybe for the purpose of advertising. And they even give out grants to study aging-related disease. So again, I don't know what their products are, but they call themselves an aging company. But this is, of all these companies that Altos is a startup as well, but Gordian is the smallest kind of startups to study aging-related. Because aging, as you can imagine, is a pretty big topic. You can't really just study aging with a startup, so it'd be hard. So there are other interdisciplinary effort, I called it, and this is where I work in. And I will talk a little bit later about this organization. But long story short, that again this is a laboratory for genomics research. It was fully funded by GSK, and then they want to leveraging Jennifer Doudna. And basically back then it was a Jonathan Weissman. He was still here, but now he moved to MIT. And so basically they want to leveraging CRISPR technology to really study all disease possible, and in collaboration with UCSF and UC Berkeley of a faculty. And there is this innovative genomic institute at UC Berkeley. So these institutes that they hire a lot of scientists. So these are not just normal academic research labs, that they would hire these scientists to tackle some of the most challenging questions. For example, back then in COVID, UC Berkeley has this effort in IGI. And then 23andMe, if you've done genetic testing, that this is a product, and they have a lot of leverage in terms of genetics and genome analysis, and they've been collaborating with GSK. And then Recursion co-founder back then was Ann Carpenter at Harvard. And then now that they really spin out from a startup all the way, and now that they have a beautiful campus at Utah, at the Park City. So apparently they left the Bay Area, probably because of the high expenses, but they still try to keep their connection with Genentech. So there's a lot of collaboration there. And then, of course, that the BioHub, which is the Chang Zuckerberg initiative, and then they started the BioHub, and it's their own entity that they try to collaborate with Stanford, UC Berkeley, and UCSF faculty as well. And the most recent ARC Institute, this was co-started with Savannah Corman, with Patrick Su. Patrick Su used to work with Feng Zhang, and then there was another faculty at Stanford as well. So it was this three faculty led of a institute that they want to study neurodegenerative disease or neurological disorder. So they have endowment by Stripe, basically the tech company, that it's a secured endowment, so they can start, and they're building new campuses. And this is another type that they want to do something that is academic related, but they want to do something and they hire an army of lawyers. I think in the hope of eventually, if something interesting comes out, they want to be able to patent it and immediately turn into products. So these are basically the things that I know. The last but not the least, so this is my least knowledge. In terms of device, because when we think about device, again, that from both Stanford and UCSF, that there are some devices that can be used and innovative. So when I was at Stanford, my friend Christoph Leutzer, so he's working on augmented reality. So again, as you can imagine, it's borderline tech. But again, his idea is how can we, kind of use for patient's diagnostic? Would it be better that if you can look at your own fMRI, you can walk into your own fMRI? Wouldn't that be better? Wouldn't it be better for you to look inside of your brain and to see where are the pathology comes? Wouldn't it be better for the doctors to go inside? So that's what the augmented reality. So the company name is called Nakamir. And then this company seems to be doing well, and he's been giving talks. So I got this from one of his posters of the talk. And this was just published yesterday, and at UCSF, Eddie Chang. So as we know that a lot of these patients, including Stephen Hawkins, right? Like when you have ALS, that your motor systems start to degrade, and eventually you lose the capability of the speech. And then there are these neuroprosthetics, and they're able to decode what you want to say, and then they will project on an avatar and can speak for you. Instead of those robotics voice, it will be a real avatar talking. So that's definitely is an improvement. So I would imagine that Eddie probably has companies in dealing with this, and I'm sure that this is an effort for a lot of people as well. So this was just released yesterday, going after the peer revision, and it was published in Nature. So again, that these devices like this, that it might be something to invest in if there are companies to look for. So yeah, and then I want to point out a couple of things in case that there are people who really want to go into. So there are conferences that you can go if you're really interested. I go to some of these conferences, let me know if you want to join, and then I can introduce you to people, and we can definitely kind of mix with them and talk with people on things. So the first one every year, and for the next year, will be January the 8th until the 11th at our very San Francisco. So that's why that you can go. It's at the Francis Hotel in the Westin. It's downtown San Francisco. JP Morgan Healthcare Conference is the whole world. All the people, they will go. Pfizer, Merck, Novartis, AstraZeneca, you name it, Amgen, you just name it, like all the people will be there, and then all the high profile people will be there. It's the new drug targets indicator, new technology indicator, and it's the social mixer of the scientists, of the venture, and for me, that the limited venture I know, Sequoia, OrbiMed, and then it was my downstairs is a Third Rock, if you know that venture company, and again, like everybody will be there. So this is like this is almost like the Oscar of the healthcare. And Society for Neuroscience is the one that I go. By the way, that JP Morgan Healthcare, I think they projected every year is a 14,000 attendance. So you just know it's just huge. And then for Society for Neuroscience, it's a 40,000. It's the largest neuroscience-related conferences. And then right now, they're so big. There are only 4 cities in the US can host this conference. Louisiana and San Diego, San Francisco, and DC dropped out. And right now, it's Chicago. So nowhere else can host this conference anymore because it gets so big. So again, that this conference is so big that you can go for science, neuroscience education, pediatric, neurodegenerative, computational. So Google go to these conferences as well. AI, ML, drug targets. So any topics you can think of, Society for Neuroscience will be one of them. And then the one I also go is called the International Society for Stem Cell Research. So this is one of the smaller ones. So you can see that I'm going from larger to smaller to selective kind of, the larger the conference, unless you have somebody you really need to go, or if you're really out of the field, you want to get into the field, larger conference is the way to go. But if you really want to meet people, you want to talk with them, have in-depth conversation, you should go to smaller ones. This ISSCR, this conference is, again, that all these stem cell related work, like media making, new technology, new induction, xenotransplant, immunotherapy, that all the people will go to that one. Keystone Symposium, this is really small. It's very exclusive. Every year that there are only a handful of sponsor, like if you're sponsoring, so Third Rock goes to this conference, they sponsor this. Genentech does, Biogen sponsor this conference. Novartis, they do, and again, you will see representations of these companies. There are panels for, where are we on disease treatment? And I go to Keystone Symposium every year. And again, it's very secluded. It's in the middle of nowhere in Colorado, Keystone. And sometimes they're in Canada, Banff. So again, it's like these locations in the middle of nowhere. The only thing you can do is networking and to talk with them. So, so that's a great opportunity as well. And let me know if you're interested in going. Cold Spring Harbor is in New York, and this is another small conferences. So as you can see, that Keystone is at the middle of the country, and if people who are in Boston, Europe, or maybe New York, that a lot of people go to Cold Spring Harbor. But of course, West Coast like us, UCLA, UCSD, UCSF, that we still go to Cold Spring Harbor from time to time. Other resources, if you really want to get into biotech investment, there are 3 websites that, I pay attention to. I see things. It's called Biospace, and there's another one called the Fierce Biotech. Who is getting a layoff right now? Biogen had layoffs, thousands of layoffs, and these are all we will know first. And then you will see some of the companies are not projected very well that you'll be able to see. And again, I follow these, and whose phase 3 is failed. So these resources that they will tell you. And STAT plus, STAT normal is fine, but STAT plus, there are experts in the field that they will write commentary. And that is the plus. So it's a subscription based. You're able to get some insider opinion. Like, you will know way before, like Lilly is gonna do very well this year. Buy something from them. Or like Lilly's projected to acquire 3 more companies, meaning that their portfolio is gonna be so much better in neurodegenerative disease. That will be something that, you can get from STAT plus. Yeah, and again, please reach out if you have any questions and you want to continue chatting, for coffee and if you're I really enjoy sharing with people on neuroscience, genome editing, or iPSCs. So if you just want to talk, I'm very interested in, talking with people on these things. And please just reach out. My email address and then my LinkedIn's appearance is marcus.h.lee. So please feel free to reach out if you have any questions. But I'm happy to take any questions now as well. Thank you. Yeah, sure.
About the future of drug delivery, LNP versus AAV, antivirus. The delivery mechanism that's most amenable to sort of the next generation of therapeutics, the targeting, or the size of the therapeutic, or the mechanism of therapeutic.
Yeah. So we have on-site audience. So what Adam was asking is, so again, there are different type of viruses, like adeno-associated virus, and I'm using HIV-related virus, which is lentivirus. And again, now that there are a lot of deliveries like a viral, like particles, or some cholesterol-based, that it's kind of easy to go in. Again, all we want is to go through the blood-brain barrier. So all these have all kinds of advantages. AAV, you can deliver big constructs, and that's why AAV is better in that sense. If you want to deliver a gene locally onto a specific cell type, AAV is the way to go. So there are companies, for example, I think Sangamo is working on AAV toxicity and the dosing, and And again, if you want to do efficient or smaller, and you may turn into lentivirus. And again, that right now, what Genentech is doing is this viral-like particles or some small vesicles that can carry your payload or your interest and can go in. So again, it's kind of a racing to see. And also, I think that it depends on what kind of disease, right, you want to treat. If it's peripheral, let's try all of them, see which one works. But for central nervous system, it's just so many checkpoints that we can't just, just go. Unless that it's a terminal disease, I guess that FDA will have a better, a more tolerating. So yeah, but thank you for your question.
What about vaccines? Typically toxin, which is kind of a different area.
Yeah.
Is that active in the Bay Area, or the companies working on various other area things that they can protect us from?
Yes. Well, Michael on site is asking about the vaccine. I can only touch on a little bit because again, it's not my area of expertise. So again, right, as we see, really that the race really started with the traditional vaccination, right? Like the Chinese, the Sinopharm or something, Sinopharma or Sinopac. No, Sinopac is for petroleum. Sinopharma, something, Chinese. And again, the traditional way of making vaccine is you're using dead virus capsids. That's the traditional way. Very efficient on most of the virus. But apparently that for the COVID, it doesn't seem to work well. And that's when the BioNTech with Pfizer, that's when it really take off. And then in the Bay Area is the Moderna with the David Rossi, maybe that's his name. And again, like that was the way that messenger RNA start to be taken into consideration. And now that it seems to be working on COVID, now can we expand it to other, right? And again, that AstraZeneca is working on it, and then GSK maybe a little bit late that is working on it, and now that it draws a new wave of innovative vaccine, and there are proprietary elements in Moderna, whatever that they're having, that it seems to be increasing the uptake of the messenger RNA. So again, as I said at the very beginning, I think that scientists will continue to think about what will be the next generation of vaccine, right? The dead virus capsids, now that it's evolved into messenger RNA, what will be the next one? And I guess that it will still be, again, the biggest drawback for BioNTech, Pfizer is the storage, right? It's back then, if you hear the news, it was really difficult to be transported, right? Because it needs a little bit lower than -20. And again, right, there's so many things that to consider in terms of making vaccine, right? And not just the safety and the efficacy of it. So hopefully that we can continuing kind of coming up with different idea. And I would assume that, the next generation vaccine will be something that, maybe it's the next Moderna that we never heard about the company. It's just a new mechanism. And to answer your question, Bay Area, definitely, like East Coast, West Coast, definitely there are people working on vaccine. And GSK is working on the HPV vaccine, newer vaccine, and some rare viral infection of the vaccine that I know. AbbVie, of course. So again, like we're not a lack of companies are continually working on vaccine, but we just need some new ideas of coming up with the next generation. So that's kind of what we're working on. Yeah, and again, I think that messenger RNA will continue getting attention because again, it seems to be working. I was skeptical because messenger RNAs are hard to deliver. They can be degraded because we have defense systems. So again, like the way, and then I was really impressed. Like it's working. This is great. And again, right, that we are also yet to see, by introducing messenger RNA to, the messenger RNA of the virus, like or the COVID virus, like what are we doing to our own immune system, right? Like are we going to eventually losing the immunity? Because right now we're having these dosing, with the traditional cooked vaccine or the dead capsids, we never need to do well, I guess we do it less frequent, not like every 6 months, right? Because we do it like every year for like influenza vaccine. But again, for a lot of these, I assume perhaps the herpesvirus, it's like you do it and it's good for a while. So again, that, we want to have something that we don't have to take every so often. It's so efficient, easy to transport, highly accessible, and all these things together that hopefully there's a new vaccine coming up.
Thank you so much for your presentation. I was wondering if you could maybe, tell us, kind of in general, why you choose lenti versus AAV, which is more popular choice, and what you thought of like LNP in terms of like applicability in the central nervous system.
Yeah, so sorry, what's your name?
My name is Jennifer. Nice to meet you.
Likewise. So Jennifer on site was asking again that coming back to the issue of, we have different deliveries. So AAV being adenovirus, and then they can carry, so they are almost like like a train. So they can carry larger cargo to go into the destination. Lentivirus is a little bit like perhaps like a plane that it can deliver. But again, that they need a lot of things, residual things like helping the cells to take the virus, and this itself is perhaps toxic to the body. And I know at the cellular level that it's really toxic for taking in lentivirus, and you almost have to let the cells recover even before treating the disease, just by delivering the virus, that you're actually bombarding the cells. Because again, we don't, as a human, that we develop the immune system, we just don't like anything foreign. And there are these rejection, and therefore is this immunosuppressants. So we have all these things. So as I said before, that adenovirus being good of delivering larger payload or larger things like genes or even protein, you can deliver protein inside with a viral particle, viral-like particle. And with AAV, you can deliver the gene transcripts, like the actual messenger RNA in through AAV. But lentivirus, you can only deliver something small. But again, it's very efficient. So again, it's like a plane. You can deliver something, but it's just don't expect it to be larger volume. But to a certain degree, like maybe a small volume is not going to do the job. Even it's efficient, it's not going to do the job. But on the other hand, with AAV, you can deliver a lot of things, but it might be toxic. So, there's this constant of a try and errors. But also the other thing is the human brain is just an amazing place that, some of the delivery, it works very well, but other genes, for some reason, it's just not working well. And that's why that, compare with viral delivery, and now they're using synthesized viral-like particle. So they're not virus, they just look like a virus, the structure like a virus, but they carry something else. So it looks like a hybrid of a train or a plane. It's a little bit faster than the train, and it's a little bit higher load, maybe a shuttle, and then to shuffle into your brain. So again, like this is such a hot field currently in biotech, and how to efficiently deliver things across blood-brain barrier in. So yeah, so you're asking a company question. So there are multiple companies, again, including Denali. They're working on their particles. How can they deliver it in?
Tell us a little bit of iPSC-derived organoid that you're working on.
Yeah.
I used to work on iPSCs as well with cardiomonocytes.
Yeah.
So my question would be, a lot of times the iPSCs may or may not have the genome profile that you have, because iPSCs are real cells, but you're differentiating them and then differentiating it into a specific cell type. So you may not have the genome that you wanted. So do you specifically select some of these cells in order to differentiate into the neural, to build the models that you want? And then, what's your selection process? And then, the delivery, these are brain organoids, so you're not really having the blood-brain barrier issues. So what are the pros and cons of using an organoid that you have since far?
Yeah. So Jennifer's question is, she's asking again related to the things that I work on, which is iPS-induced cell type into cerebral cortical organoids. So related to in terms of the protocols and also I guess it's how can we use that to shed the light on drug discovery. So these are cerebral organoids that you give them sensing cues, like what you do with the KIR on the iPS-induced cardiomyocytes. I also know how to do that. I work with Bruce Coughlin at Gladstone. So basically that you add these cytokines or the cues, let the cell clusters develop into brain, just mimicking the brain development. And when we use iPSCs, as you said, so there's this conversion from, let's say, from me, a 30-year-old of a person, if I do the iPSC conversion, that we call it epigenetic signature. So the 30-year-old signature is gone. And basically that it's a 0-year-old of a cell. So again, a lot of these epigenetic signatures seems to get lost. And the way how people deal with that is currently that people can use fibroblast, which is the somatic cells, directly bypassing the iPSCs, directly inducing into organoid. It may or may not be very efficient, but in that sense, if you study aging-related disease or personal medicine, like specifically you want to study just you, like the now you disease, then you can directly convert from somatic cells to the organoid. So you can do that. And specifically that the most recent, I came back from this conference, again, it's the functional genomics and brain development. This is a Gordon Research Conference. Rusty Gage at Salk Institute, he studies schizophrenia. He directly did patient somatic cells inducing into neuron, capture the epigenetic signature, study disease in that way. So we can do that. It's just not super well established. And again, the way how you strip down into iPSC and then to the cerebral organoids, this is a better study or better characterized. So yeah, and again, that how do we use the cerebral organoids to study, blood-brain barrier, because they don't have blood-brain barrier just yet. And again, that's why that Li Wei Cai, she's at MIT Pickover Institute. So she discovered iPS-induced, she called it induced blood-brain barrier called IBBB. So what she does is to use these, we call it sandwich or pancake cell culture. So you would have the cells making up of the blood-brain barrier, induce them separately and combine them together. And then in that case, you can model, like the things going through blood-brain barrier. But at the end of the day, it's still different, right? Because her IBBB is flat. So you want it to be with the curvature and everything. So her former postdoc, his name is Joe Blanchard, and he moved to Mount Sinai, has his own lab, and I hope that he's going to carry, because that's what he was originally doing this, and then he brought his research program to Mount Sinai, and hopefully that he can come up with something that can better model IBBB, and then in order to study how can drug penetrate. So that was one way. And the way that I study this is I don't use it to study, drug efficacy, but what I use to study is how can I model, currently I'm using a brain tumor. So I'm using pediatric brain tumor. I want to study how do tumor interact with cerebral tissue. So that's what I use it for, because tumor cells for some reason can go through blood-brain barrier, right? That's why there are these peripheral cancer, breast cancer, lung cancer can metastasize into the brain. Like, we don't know how do they do it, right? Because again, you would think that cell can be blocked outside, but they didn't. So again, like I can study these type of metastasis. That's how I use the cerebral organoids for. So again, it's just like you have to, like every organisms, right? Philosophers say that every model system has their own flaws. You just have to understand how to use it. And then of course that, eventually that I think that we need to, the people who study using mouse, there's still value to use mouse because they have in vivo or they have the in the body. But again, everything I do is in vitro, is outside the body. I'm doing cell-based assay. So again, we want to, work on these things together, so we can, look at it outside and inside the body before we move on to human trials, right? So, yeah, of course. I hope that this is helpful for everyone, because we are at 6:30. Is there any more questions at the Zoom?
Yes, hi, can you hear me?
Yeah, we can hear you.
Yes, I have a quick question about the AI for drug discovery companies, and I think you probably covered some of it already, but I just wonder if you know like a survey of the best, or what you think of the best approaches or companies that seems the most promising. And I'm particularly kind of more interested in the small molecule structure predictions, but there's so much more involved in this process. And I wonder, everyone's talking about using AI for drug discovery, and I just wonder what part of that process is the bottleneck that would be the most useful to work on, and which one seems the most promising in terms of leading to discovery.
I see. Thank you for your question. This is a little bit outside of my field, but as far as I know, that AlphaFold by Google, that was one of the very well predicted crystal structure. So back then, every crystal structure seems to be a major discovery back then. And that's why that when Brian Kobilka from Stanford discovered the G protein couple receptor, it's like the major discovery because it's so hard to look at the structure. It's kind of like you have to know the structure, therefore you see the binding pockets and you see the interaction. So I think that a lot of AI ML lab, like including the AlphaFold, they're trying to predict that when you put in a small molecule, and then you want to see how does it change conformationally. Now, that's when the barrier comes. A lot of these small molecules that are developed by drug companies, it's not public.
Yeah.
And you may have difficulties of getting first-hand information of those, there, let alone of shooting out the crystal structure. And if you don't know the crystal structure, how can you know the binding pocket? How can you know the specificity? And what currently that I'm seeing is there is a company, it's by UCSF, again, Brian Kobilka's former postdoc, and his name is Uwe Klein, that he was at the Numerate. Numerate is kind of like a little bit like an AI ML company. They try to predict the structures of compound or small drugs. How do they interact with receptors, like an interaction, and how do they change? And now that if you change one more amino acids, is the conformational change is gonna be different? Are you gonna acquire a new function? So these, you cannot do point mutation one by one for these experiments. That's when AI ML comes. And other directions, if it's a small molecules, that I think I was at this Icon Therapeutics. The person was from Recursion, so he knows a lot of imaging, and now that they're working on single molecule tracking. So if you can, track a single molecule, then you can see the protein, and perhaps that if that protein binds specific compound that they can change, and then you can study. And again, that will be array-based of a screen. It's so large of a scale, and it's such a such a big analysis, and you have to use AI ML. So these are the two directions that I can see companies are using AI ML to advance their research, and kind of like incorporating with a bunch of scientists, because with a lot of the questions that you can ask with AI ML, like may or may not translate in real life, and that's why that there's value for AI ML as an initial screen, that if you see the target seems to be working well, then you focus on these several amino acids mutations, or these several drug compounds. That's kind of how I can see.
One of the companies that I was looking at was something by Daphne Kohler with In Sitro, and also Michael Levitt, and they're both from Stanford, former Stanford professors. And I just wonder, do they focus on this on these particular small molecules, or are they looking at it more from an end-to-end perspective? And because they seem to be doing something quite different and using computer vision.
Yeah.
part of it.
As I mentioned earlier, that in vitro by Daphne Kohler, basically what they're doing now is, they have three arms. The first is technology development. So in vitro is really trying to develop, like a new technologies in terms of, again, AI ML. How can we predict the drug behavior, and how can we predict that exactly what they do? I'm not 100%. And in the middle, which is they are developing CRISPR screens. So they're looking at the pooled CRISPR screen, and they're array-based screen, and there's also optical pool screen, firstly developed by Paul Blaney at Harvard. There are a lot of people from Paul Blaney's lab joined in vitro, and in vitro is really trying to use morphology as the readout and to perturb specific phenomenon and specific, they are trying to get gene targets. And then the other thing that in vitro is working on is how can you integrate larger data sets across experimentally? So again, can you integrate transcriptomic data with the RNA-seq data together and with CRISPR screen together? So these, you can say that it's also borderline of a AI ML, because of course that you need machine learning to do these type of things. So these are the things that in vitro is working on. What was the other company you were saying?
Can I follow up on that thought? Because what you just said in the last part is super interesting. Like I also talked to NVIDIA a little bit, and I wonder if there's a way to integrate all of that to do something similar to what they're saying, like using something like generative AI or just large, large molecule models or large gene model to actually integrate all of those together. And if you see any progress in machine learning toward that one kind of almost unifying model where, one model, many use cases, and then do fine tuning. Yeah.
I tell you where the bottleneck is. The bottleneck is that all these data are in different forms. Each one of them tens of hundreds of gigabytes, and then you need a supercomputer to convert them into the integrated database. That's when most of the effort comes. Because during that process, that a lot of data either get lost in translation, just unconvertible, and then a lot of the data sets may not even be public, because you need a lot of larger data sets to do the machine learning, right? Because otherwise, what can you learn from? So that is a major bottleneck. So my assumption is that you have to have a lot of experimentalists to generate the data for you or for the machine learning computational biologist to train the data sets. So what I want to say is these probably require big pharma company and collaborations across labs to do AI ML.
Okay, that's amazing. Thank you so much for that answer.
Yeah, of course. And again, that I'm no expert on that, but I'm just, I have my own computational biologist, and I like her to look at, hey, could you take a look at this paper that their microglia gene signature with the other paper of the microglia gene signature? I was thinking it's 5 minutes of a work, but she told me like, you're just gonna wait because this data set is not compatible. I cannot compare this with this, and I need to convert it, and the computer crashed, and that's not working. This is not working. Give me 2 weeks. And then after 2 weeks, she gave me 2 dots and say, they're similar. And
that's super interesting. Yeah, because I work on models, on models in neurodegenerative diseases too, and I look at that problem and also, so I'll definitely follow up, and thank you.
Yeah, exactly. And again, like if you want to do AI ML, I think that, these nonprofit organizations, ARC Institute, Stanford, like IGI, like us, Laboratory for Genomics Research, like all these places, I think they will be a good place for you, because in that case that we have enough of the data sets for you to learn, to train. Otherwise, you just have one data set. Every day, you're probably just compiling extra data sets,
I'll definitely, thank you so much for that. I work with like large pharma, so I have access to quite a large library of compounds. So, and then, so we're trying to figure out what to do with it, to be honest. And this could be like a national level project for some of the groups that I'm working with. So it's outside of the US, but very interesting.
Yeah, exactly. That's why that GSK is collaborating with 23andMe and on the GWAS, and again that Recursion's working with Genentech, and GSK is working with UCSF, UC Berkeley. So you need these collaborations, and that's why. And pharma that there are collaborations, right? That's why the 23andMe sold
Yes.
their genomic data to GSK for 5 years for them to mine.
Yeah.
It, right?
I'm not sure how good the data sets or how useful the 23andMe data is actually, because it's it's not even whole genome sequencing, but
yeah.
they may have overpaid, but it's interesting.
At least that it's a same format of data, as I mentioned, right? At least it's the same format, and also me has the largest non-white population of the genome information. Because again, right, when we talk about GWAS, it's all Caucasian. But what about Latinx? What about Asian? Like we have no idea. Whereas 23andMe has all of these data sets. And of course, that the UK is trying to increase, and then Sanger Institute at the UK is trying to.
The UK Biobank.
UK Biobank is trying to get more of those data, but again, not
their data set is also not clean. Yeah. This is super. I wish I was there in person. Thank you for this.
Yeah. But again, you feel free to follow up with me anytime. And again, that I'm always happy to chat more. So
that's so amazing. Thank you so much.
Yeah.
I'll definitely follow up.
Sure.